Hello, my name is Richard Gantt, and I'm a senior staff scientist here at Twist Bioscience. I'm here today to talk about how our universal blockers, universal adapter system, and our development efforts at providing large sets of qualified, unique dual indices are laying the groundwork to provide a robust and high-performing multiplex target enrichment workflow for our customers. As we talk through this poster, I encourage you to pause the video and read through the finer details that are provided in each pane and in the figure descriptions. We firmly believe here at Twist that our best customer is an educated one. Now, early on during the development of Twist target enrichment system, we were heavily focused on probe design and hybridization performance. However, it didn't take us very long to recognize that there were some serious bottlenecks that needed to be addressed if Twist was going to be able to bring a world-class target enrichment workflow that could take our customers from sample to sequencer. And we wanted to highlight essentially four key areas that we focused in on for development. First is the development of universal blockers to eliminate non-desired adapter interactions during target enrichment. The second is improving adapter ligation, and we address this issue with the development of our universal adapter system. Third is the performance during multiplex to reduce hands-on time and reagent usage. And finally, which is the main focus of this poster, is our development efforts towards increasing the number of adapter indices, or barcodes, that are available to our customers to continue to drive down cost per sample for sequencing. Now, as I mentioned, the focus of today's talk will be around that. Around the efforts that we have put forth thus far to increase the total number of available barcodes. Now, our efforts here include sophisticated index design that is context specific, making sure we're subsetting those index pairs into bins that are both base and color channel balanced. So performance is equivalent across multiple types of sequencing chemistries, and also going through the exercise of empirical validation of each index pair across multiple sequencing platform and in different contexts. Taken together, the efforts that we have put into these areas allow us to offer our customers a target enrichment experience that removes numerous bottlenecks and reduces turnaround time while still delivering high performance sequencing results. Now, the first of these areas that we'll be discussing today are Twist's universal blockers that are compatible with TrueSeq adapters. Now, these are a really nice product because these adapters are barcode independent and able to offer on-target rates during target enrichment performance that are consistent across an extreme variety of indexed adapter designs. Now, this performance holds up regardless of whether the system is either single or dual indexed. Index length from 6, as you can see in the figure here, from 6 base pair all the way up to 14 base pair, or if there is a UMI present within the adapter design. Now, with our competitors' adapter blocker products, you're more than likely going to be locked into one or two of these designs unless you work towards and order a custom product. Now, this can get prohibitively expensive, especially if you need to go to scale for larger projects or switch index design or length in the middle of a project for technical reasons. Now, with Twist universal blockers, this is simply a non-concern. You can utilize your research funds in a more efficient manner. Now, the fact that these universal blockers are barcode independent also means that you can multiplex samples without fear of losing performance. And you can see this essentially here in figure 2.2 in the bottom portion of this panel. Now, another interesting customer experience that's been reported to us with core sequencing facilities is that perform target enrichment is that sometimes they don't have control over the adapter design that the customers might be utilizing if the customer is generating their own NGS libraries. Now, because this product performs independent of index design, this simply means that they can intake samples and put them through Twist target enrichment workflow without fear of poor target enrichment performance. And this is And here in figure 2.2, we demonstrate that Twist universal blockers essentially do a great job of increasing the fraction of on-target bases across a wide variety of panel sizes. Now, it's important to note that the fraction on-target performance can be driven by panel design. Now, in this study, a number of these panels were first passed at the time and not fully optimized for performance. However, it's really clear compared to the negative control here that Twist universal blockers provide a dramatic increase in the number of on-target bases. And now on to our second area where we focused a significant portion of our development effort into improvement, and that's around the ligation efficiency. Now, we recognized early on that ligation during the NGS library creation process had a huge impact on the quantity of NGS library that's available to move into the next step of the target enrichment workflow, which is in our case, which is hybridization. Now, in addition to addressing quality with industry standard full-length Y adapters, there's an undesired presence of adapter dimer that can artificially skew the quantification of the final NGS library and can actually result in the customer underloading their libraries when pooling for hybridization. Now, this has a significant impact on our customers who are running hundreds or even thousands of samples every day. It's something we wanted to address during our development efforts. Now, the manner in which we attacked this problem around ligation efficiency was drastically changing our initial adapter design. Now, instead of the industry standard long Y-shaped adapters that contain indices that are unique for each sample, Twist's universal adapter system consists of a shortened universal adapter that is identical for all samples. Now, the process of index installation is then shifted into a downstream PCR step that is typically much easier for an individual researcher to troubleshoot and also control. Now, as you can see in these figures, the results of these changes are dramatic for both the total observed concentration and absence of adapter dimer. Now, the samples here were generated with full-length adapters in this short study were subjected to 10 cycles of PCR, and while the samples generated via Twist universal adapters and 10 base pair unique dual index primers were subjected to 8 cycles. Even with a decrease in the number of PCR cycles, we observed nearly a doubling of the final observed concentration and therefore the total yield. Now, this reduction in PCR cycles helps to improve data quality by reducing the starting number of PCR duplicates that are subject to target enrichment. Another distinct advantage that Twist's universal adapter system provides is the absence of adapter dimer formation. Now, adapter dimer formation has been a real sore spot in NGS library creation. When free adapters find each other in solution because of the nature of the ligation reaction, they simply ligate to each other and can be utilized as a PCR template during the next amplification step during NGS library creation. Now, due to the reduced size of Twist's universal adapters, adapter dimer formation is greatly minimized, and any dimer that would form is so small that it's simply removed during the post-PCR cleanup step during NGS library preparation. The major advantage here is that you get a much more accurate quantitation of your final NGS library. Now, as a result, when you go to pool your NGS libraries for hybridization, you can be confident that your samples are evenly represented and you're less likely to have dropouts or samples that underperform or overperform during sequencing. Our customers report that they love the system not only for target enrichment, but also for whole genome sequencing, where adapter dimers can consume a large percentage of reads if they are present in high molar ratios when compared to the desired genomic material that is being prepared for sequencing. Now, taking the two areas that I just discussed, which is adapter blockers and universal adapters, we can now demonstrate that we can use these products actually in concert and demonstrate great multiplexing performance across a wide variety of different panel sizes. Now, on this graph, we actually have 3 different panel sizes representing 0.8, 3.3, and 33.3 megabase of targeted sequence, and 3 different multiplex pools with that either contain a single sample, 8 samples, or 16 samples within their multiplex pools. Now, all of these pools are subjected to target enrichment with our standard target enrichment reagents with a 16-hour hybridization time. Now, as you can see in the figure here, we have very little drop in performance for the percentage of bases observed over 30 times when moving from a single isolated sample to a sample with either 8 or 16 samples present within the multiplex hybridization. Now, being able to plex at these levels allows you to greatly limit the number of hybridizations that need to be executed for a given number of samples, meaning that you can focus more attention on fewer captures to ensure your technique is more consistent. Now, in the context of automation, this actually means you can greatly reduce tip and plastic costs due to the decreased number of hybridizations required for large sets of samples. Now, even with this great multiplexing performance, we weren't satisfied here at Twist, and we knew that the next bottleneck for customers was going to be around actually getting more samples on per sequencing run. The number one way Twist was going to be able to enable our customers here was to continue to drive down cost, was by greatly increasing the actual commercially available number of unique dual indices. Now, in this next panel, we'll be discussing how we went about increasing the development that we went about around towards increasing the total number of adapter indices available to our customers. As you're probably keenly aware, although the amount of data generated per sequencing run has continued to increase almost exponentially year over year, the number of samples that can be analyzed per run has continued to be artificially limited due to the lack of large and high-performing unique dual index sets. Now, one limiting factor in the identification of such UDI sets has really been the lack of cost-effective workflows to generate thousands of NGS libraries with a representative genomic insert. Now, to address this limitation, a high-throughput NGS library generation method was developed here. Now, this uses a combination of fragmentation of a single human genomic source and automation in concert to generate thousands of NGS libraries in a cost-effective manner in a method through a method that we refer to as the creation of artificial barcoded libraries. Now, here we have a high-level overview of this workflow demonstrated through our electropherograms represented at different points in the process. Now, in A, we have a PCR amplified ligation product that is very heteroduplex and difficult, complex and difficult to quantify. In B, we're actually able to polish this product via PCR, and then in C, install UDI primers for actual evaluation. Now, when compared to a typical benchtop NGS library, which is displayed in D, the resulting size and distribution are nearly identical. Now, as a result, we actually have a system in-house here at Twist that is capable of generating thousands of NGS libraries with representative genomic material as insert for screening of UDI performance. Now, the presence of human genomic material as insert is actually really critical to ensure that any bias that would be observed during NGS library creation is actually equally represented throughout the entire population. Now, the next area that I'll be talking about here, where we have put a lot of thought and effort, is around the algorithms that are utilized for both barcode design and also subsetting sets of barcodes into usable sets for our customers. Now, anyone who's gone through the exercise of developing index sets will immediately recognize that designing these algorithms to demonstrate unique dual indices at scale that also remain both base balanced for multiple chemistries is basically a non-trivial exercise. Now at Twist, we kind of have gone through this development process for large UDI sets with kind of three major things in mind. First is sophisticated index design that is actually context specific. The second is subsetting index pairs into bins that are both base and color channel balanced. And three, the actual empirical validation of each index pair across multiple sequencing platforms that is enabled by our artificial barcode library system that we just discussed. Now, for the first point, the design, parameters such as minimal Hamming distance, the elimination of homopolymers, and also longer repetitive elements, hairpin formation, the percent GC content, the avoidance of multiple dark bases at the beginning of each index to address limitations of the underlying sequencing chemistries, and also a number of additional other rules are utilized to design large pools of unique dual indices that are likely candidates for UDI primer sets that have even and robust performance. Now, to address subsetting, we actually take into account melting temperature, reverse complement matches within a proposed subset, base composition at each index, and also check on color balance, color channel balance at each position. Now, in our figures here, we actually have examples of the algorithms that provide a stark difference between color channel balance for 2 and 4 color chemistries that we can see in figures A through D here. Now, figures A and B essentially demonstrate a unoptimized algorithm that provides a single index set with excellent performance for 2 color sequencing chemistry, but is completely unbalanced for 4 color sequencing chemistry, especially as you get towards the end of the index there. Now, for figure C and D, this demonstrates a second optimized algorithm, which is actually able to provide single index sets that are much more likely to be appropriate for both 2 and 4 color sequencing chemistries. Now, algorithms capable of both generating and subsetting UDI pairs balanced across multiple sequencing chemistries are really critical to robust performance. And in terms of the empirical validation that we do, that's addressed through both the artificial barcode libraries that I just discussed in the prior pane, and we'll also provide some examples here in the last panel. Now, in this final panel, we'll be discussing about how we went about increasing the number of adapter indices available to our customers. Now, as you're probably keenly aware, although the amount of data generated per sequencing run has continued to increase, the number of samples that can be analyzed per run has been artificially limited due to the lack of high-performing UDI index sets. So one limiting factor in the identification of these UDI sets has been the lack of cost-effective workflows to generate thousands of NGS libraries with a representative genomic insert. And as we just discussed in a couple of panels ago, we've gone through the exercise of generating artificial barcode libraries. And this is now a system that where we can generate thousands of NGS libraries with a representative genomics material that acts as an insert for screening for UDI performance. And as we mentioned previously, the presence of this human genomic material as insert is really critical to ensure that any bias during the NGS library creation is actually equally represented throughout the entire population. Now, using this approach, we essentially generated just over 1,000 NGS libraries containing unique dual sequences that were constructed and screened in an iterative fashion for even sequencing performance. Now, our initial screening effort with over 1,000 pairs of these UDIs is presented in the figures that you can see here as histograms, where the sequencing performance was determined by the number of observed reads and is centered around an arbitrary value of 1. Now, it might be a little hard to see here, but out at the tails of the histogram, we have a number of over and underperformers that are either not generating enough reads or are being overrepresented when compared to the majority of the population. Now, as we move through multiple rounds of screening, we're actually able to eliminate over and underperformers and greatly tighten up the distribution that we just observed in figure A here. And this is demonstrated in figure B, where we have a total of 384 UDI pairs with a sequencing performance that is plus minus 25% to the mean for all members of the population. Now, this metric very nicely holds up when we break this population out further into sets of 96 here that are presented in panels C1 through C4. Now, as a result, our customers can be really confident when they go to sequence large numbers of samples. The likelihood of samples either being under or over sequenced is greatly reduced, and you have a much more consistent representation of samples in terms of the number of reads observed. And this is key to cutting down on the number of repeats that one would need to do during the course of a experimental series. Now, while this specific investigation identified a set of UDIs suitable for high multiplex hybridization applications and investigation, as you can see, this clearly lays the groundwork for identification of much larger UDI sets, and those are ongoing efforts here at Twist. Now, here at Twist, we've undertaken a systematic approach to developing systems that are required for identification of large UDI sets and are currently undergoing efforts to greatly expand our current product for UDI primers. Now, we believe offering large UDI primer sets when combined with our multiplex hybridization system and high-performance target enrichment panels are going to greatly expand the accessibility and continue to decrease costs for targeted sequencing at scale. Now, today, we briefly highlighted and discussed a number of products and development efforts that help our customer eliminate bottlenecks and reach peak performance in target enrichment workflows. Now, whether your target enrichment workflow would benefit from our universal blockers to increase on-target bases, you would like to increase NGS library yield with Twist Universal Adapter System with UDI primers that are tuned for even sequencing performance, or you need a high-throughput target enrichment solution, or it might be all of the above, Twist is ready to support your research efforts with these world-class products. And look forward to additional announcements around our UDI product offerings in the near future. Thank you again for spending some time with us today. Please reach out to us through your local sales representative or at twistbioscience.com if you have any questions or would like to learn more about any of these products. Thank you.